Thanks, Matt. Thank you all for coming, and thank you, Bioconductor organizers, for letting me give this talk. My name is Shian Su. I'm a PhD student from the Ritchie Lab at the Walter Eliza Hall Institute in Melbourne. And today I'll speak to you about visualizing nanopore methylation data using a package I'm developing called NanoMethViz. So to begin, I'll just give you a brief introduction on what kind of methylation I'm looking at. So it's DNA methylation, which is the addition of a methyl group to the DNA molecules, and it's the most common form of methylation in mice and humans. These methyl groups are added to CpG motifs in the DNA, and in mice and humans, this generally has the effect that it suppresses gene expression downstream of where the methyl groups are added. And this usually happens in the promoter regions of genes. So this mechanism plays an important role in genomic imprinting, which is the expression of a gene from just one of the parents. X-inactivation, which is the process in female mammals where one of the X chromosomes is transcriptionally silent, and the suppression of repeat elements. This plays a really important role in early development of humans and mice, where misregulation leads to disorders such as Prader-Willi syndrome and Angelman syndrome. Then to study DNA methylation, we're using Oxford Nanopore sequencing. The way that this works is that we saw from the previous presentation that a motor protein pulls a DNA molecule through a nanopore here. And as that molecule passes through, electrical signal is detected and recorded. And then the original use for this technology was to detect bases. But because of the way that this technology is applied, we're directly sequencing DNA without doing PCR, which means that all the methyl groups are actually still attached to DNA. And this causes enough of a disruption in the signal that we can actually identify whether or not bases are modified as they pass through. Having these long reads gives us quite a significant advantage for the things that we're trying to study compared to previous technologies using short reads to study methylation. Because having long reads lets us have a significantly better chance of haplotyping a particular read that's figuring out which parent that transcript came, or that DNA fragment came from. And the way that works is to detect the haplotype of a read, we need our read to overlap a parental SNP. So these parental mutations where we know which parent it belongs to. And with short reads, the chances of overlapping these parental SNPs is quite low. Whereas with long reads, the chances of overlapping these parental SNPs are much higher. And this allows us to have a much higher proportion of reads that we can haplotype. In a previous study by Gigante et al. In 2019, it was found that 24% of reduced representation by sulfide sequencing reads could be haplotyped, whereas 74% of nanopore long reads could be haplotyped. And to test this technology, members of the Blurt Lab at WeHi helped design this experiment where we take F1 crosses from these black 6 and cast strain mice. These are very well-studied strains of mice and have a very well-characterized SNPs. And we've taken mice placental tissue, so female mice placental tissue. So all of the samples that we work with will have 2 X chromosomes with no X-inactivation states, because in female mice placenta, it's always the paternal, so the father's copy of the X chromosome that is inactivated. And from this data, what we have is quite a nice even coverage of the methylation patterns across the whole genome. We used 3 promethine flow cells, which is the highest throughput nanopore sequencer available. And we used 1 for each sample. This gave us around 46x coverage across the whole genome. And you can see here that the coverage is quite even, and we're getting reads from we're getting reads mapped to all regions of the genome. And that equates to around 7x coverage per sample per haplotype. And the current state of doing methylation analysis using Nanopore is you do all these steps here. You have to do base calling, alignment, methylation calling, haplotype calling, DMR detection, and visualization. And where I've put green colors in this little pipeline, so these are the steps where additional software is kind of required, because the output of one software doesn't necessarily plug into the next one very nicely. And so these little green arrows are areas where I've implemented special conversion functions in NanoMethViz to facilitate the pipeline. And down the very bottom, visualization I'm performing using NanoMethViz, which is a new package I'm developing. So as a part of NanoMethViz, I've created this format here that unifies the multiple formats produced by different methylation callers. So Nanopolish, F5C, and Megalodon. It's essentially just a simple table format, which I've compressed using TabX. And I've also produced downstream converters so I can plug my data into various analysis software available in Bioconductor for differential methylation. And as an example of what's in my package, so here is just 3 simple lines of code where I've taken a file that I've converted into my standard format. I've converted it into a BSSeq object using this BSSeq to log methylation ratio function. And then I just used the plot MDS function from Limma to create this MDS plot here. And this shows us there is good separation between the methylation patterns of our 2 different haplotypes. So the cast samples are off to the left and these black 6 samples are off to the right. Another plot I provide are these feature aggregate plots. And what these are is I've taken a big list of features. So here it's put into the function using this regions argument. And NMR is a NanoMethViz results object, which stores all the methylation information. And I've taken all of the features described in the regions argument, and I've taken an aggregate across all of them. And what's shown here is methylation across the aggregation of all the genes in the X chromosome. So using this plot, what we can see is that in the paternal, which is the inactive X, there is overall a lower level of methylation across all the genes, but slightly higher methylation relative to the maternal in just the transcription start site. And this reflects the fact that all of these genes, well, all of these genes should be silenced, and so they have slightly higher methylation in the transcription start site, although we can clearly see here that it's not a significantly higher level of methylation. And the other thing that we have to look at, so normally when we do these differential methylation analyses using the existing software, what we get out is these big tables of just areas which are differentially methylated. And because we're using a new technology, we always want to verify the validity of these results. So if you just look here, it just basically tells you, for example, in the first row, it tells you that the GNAS gene is hypermethylated in one sample relative to the other. And I guess it's a bit difficult to trust these results without seeing a visualization of it. And so the main plot in the NanoMethViz package is this spaghetti plot. And what this shows is, well, there's 2 ways to make this plot. You can use the plot gene function if you have a gene annotation and you just give it the name of the gene. Or you can plot region if you just have a region specification, so giving it the chromosome and the sign end bases. And what you get is this plot where the big thick lines in the middle are the average methylation trends across your two different groups, in this case, the cast and the black 6. And in this particular plot, what we see is that in the PEG3 gene, the cast, so the paternal samples, have a significantly lower level of methylation around the transcription start site, which is shown with the isoform annotation below the plot. And the black 6, the maternal sample, is hypermethylated, so it's more methylated in that same area. And that makes sense because it's a paternally expressed gene. And so by demethylating around the transcription start site, the paternal copy of the gene is allowed to express while the increased methylation suppresses the maternal copy of the gene. And each of the individual thin lines here show you the raw data, which is showing you individual long reads and the methylation probabilities smoothed across those. And then you've got a little shaded area in that transcription start site, which shows you where the differential methylation analysis has marked a differentially methylated region. And so these kinds of plots help you with really getting a higher resolution look into areas where your methods have marked a DMR, and hopefully they'll be very useful for researchers working with nanopore methylation data. So the current state of NanoMethViz is that I've provided and I've shown these multiple visualizations for nanopore methylation data. I've got upstream support for Nanopolish, F5C, and Megalodon, which are the most popular and officially supported methylation callers. And I've got downstream conversion support for BSSeq, DSS, and EDGAR. And currently it's under review for release in the next version of Bioconductor. And I'd just like to thank all the members of the Ritchie Lab, the Bullet Lab, and the Monash School of Econometrics and Business Statistics for helping me out with this project so far. And thank you all for coming.
Thanks, Shian, for the really nice talk. Are there any questions for Shian? Please pop them in the chat box. Good luck getting your package into Bioconductor. Are there many other packages, Shian, for long-read analysis in Bioconductor that you know of?
In Bioconductor, not in the existing release. I haven't kept an eye on the submission queue. Hopefully more people are working on this, but as far as I know, there's not much going on.
It's a question from Al. Thanks, Shian. Do you think these can also be used for single cell NMT-seq data? So that's, what's the N? Is it nucleosome, methylation, and transcriptome-seq?
I'm not familiar with that type of data, but I will go read up on it after this. And if it's applicable, I would, yeah, definitely like to support that as well.
Julie's got a question, no, a comment. TrackViewer has functions for plotting methylation data too, so that might be something to look into also.
Yeah, I mean, the main thing about this is that because it's in R, you can kind of specify and loop through a whole bunch of different areas and, you know, save those plots out. Whereas the more GUI-based solutions, you kind of have to, you know, scroll through and do screenshotting and things like that. So hopefully this is easier for analysts. Oh, I've lost sound for you, Matt.
Sorry, I didn't unmute. There's a question about, can methylation signal be used in the haplotyping?
Good question. Possibly. If there are known differences in methylation between two haplotypes, then that information definitely can be used. I'm not aware of that being done by any existing software, but that's definitely information that would be useful.
And Lei asks, how about PacBio DNA methylation data?
At the moment, I don't have any support for that, but given that the format that I use for my plot is kind of agnostic to what technology it came from, as long as you can have a table that tells you what positions are methylated and which are not, potentially you can use the package for PacBio data as well, but currently it's not something that I've looked at.
Great. Well, thanks so much, Shian.
Thanks, Matt.